Thanks for coming to, I believe, the last seminar of the fall series. So it is my delightful pleasure to introduce Professor Kristyn Masters, who is the Villas Distinguished Achievement Professor and Vice Chair of the Department of Biomedical Engineering at the University of Wisconsin. This is an even better kind of pleasure because I'm originally from Wisconsin, so it's nice to see what's going on back there. She earned her undergraduate degree from the University of Michigan, and then went down to Texas to Rice University for her PhD in chemical engineering, and since then has done quite a bit in kind of the intersection of biology, materials, microfabrication, and whatnot, as you'll see today. I won't belabor it since we'll hear all the latest and greatest shortly. But suffice it to say, Kristyn has received numerous awards over her career, both in teaching, but also both in research. And so she generally gives very nice, clear, and insightful presentations when she gives talks. So with that, I will stop taking up all the time and pass it over to Professor Masters to tell us about everything that they're doing.
Thank you so much, and thank you for the invite. I'm really excited to be able to give this seminar to you guys. So on my slide here, this right here is actually a picture of Madison. So Madison is on an isthmus. This is the Capitol building here. So we're sandwiched between two lakes, and it's a very beautiful place to live if you've never been here. Okay, so what my lab does is my lab studies how the cell interacts with its environment and the cues that are in its environment. And there are lots of different cues that can perturb cell function. And they can be everything from soluble cues, so cues that are the growth factors that are in solution, to the extracellular matrix. So this is the scaffolding that surrounds the cells and tissues, to the biophysical properties of its surroundings, including the stiffness and topography, to how many cells are near this cell. And sort of the tools that we use to look at how the microenvironment perturbs cell function are largely based upon biomaterials developments. So in other words, how can we control the presentation of these different factors to understand how individually and in combination they're affecting cell function? And so a lot of our work is premised on the concept of tissue engineering as well. And I was told this is based over a broad audience, so I'm giving you sort of a broad intro to the type of work that we're doing. But tissue engineering started out a couple decades ago and was sort of hyped as organs in a dish, where you can take cells and combine them with a scaffold material, add some stimuli to try and make the cells do what you want them to, and build some little tissues that don't actually look like these organs, but it's an idealized version of building these organs in a dish. And that's honestly how my lab started out as well, where we're trying to make these healthy tissues for healthy replacements for when people had a damaged tissue that needed repairing. But what we've transitioned to is actually diseased organs in a dish. So what my lab does is we combine the cells and the scaffolds and stimuli to make these terrible diseased-looking tissues, which you might be thinking, why would you want to do that? What use could that possibly be? And so I'll go through a little bit of how we arrived on that place of wanting to make diseased tissues and talk a little bit about this evolution of tissue engineering. So when tissue engineering first started, it was an accomplishment just to have a scaffold that supported cells that would live on it, right? So back when I first did tissue engineering, it was about 25 years ago now, and at that time, putting cells on a material and in vitro, and it's three-dimensional, and having the cells live and grow and fill up that material was sort of an amazing feat in itself. And then, we became more sophisticated in the subsequent years, and had being more intentional about engineering the environments to replicate the features of healthy tissues, to make those tissues that could serve as a replacement for a diseased or damaged tissue. But that has evolved as well, and we are starting to see value in recreating the diseased tissue environment. So not just making something that could serve as a replacement, but making something that could serve as a platform for understanding why disease happens, and allowing us to do these prospective analyses to probe the mechanisms of disease progression. So again, why do we want to make these disease models? What can we achieve by modeling disease? So one, as I alluded to on the last slide, is to understand why disease happens. There's a lot of diseases where we just don't understand the sequence of events of how that tissue became diseased. And to establish causative and not just correlative relationships. So, for many years, animals have been used as models of various diseases, and animals have lots of advantages with respect to representing the whole organism and having the immune system present and all these other factors that are really important in disease progression. But animals are also really, really hard to precisely control, and sometimes really hard to tease out causative relationships in terms of, I've made this intentional change, I'm going to see the direct effect of that. Engineered tissues could be a very useful way to identify new things to target to treat diseases, as well as to test new treatment strategies. And with respect to timeliness, it could be, one of the things that attracted me to it is that it could be a more immediate application of tissue engineering technology. So, there's some tissue engineered products on the market now, but ultimately a lot of the things that we want to tissue engineer are still ways out in the future. And this type of tissue engineering to understand disease progression could be a way where we could make a more near-term clinical impact when it comes to identifying targets and drugs. And also what we learn about making disease tissues can also help us understand what not to do when making the healthy tissues. So a lot of my talk is going to focus on the extracellular matrix. So as I mentioned before, the extracellular matrix is this sort of scaffolding that naturally surrounds cells, and cells make it and produce this extracellular matrix. The extracellular matrix, or ECM, is different in just about every tissue, has a different composition and different features, and that's part of the things that makes our different organs work the way that they do. Is this extracellular matrix enables, the functions, enables your blood vessels to, the elasticity of your blood vessels so that they can dilate and constrict, as well as your lungs can expand and contract, and your bones can withstand impact. But the extracellular matrix is also really important in disease. And so it looks different from tissue to tissue, but can also look different within the same tissue. So these two are pictures of, this is a healthy heart valve and a diseased heart valve. And these different colors in the stain represent different extracellular matrix molecules. And so you can see here that when you go from healthy to diseased, you start to get a really different looking ECM. And we're really interested in how that influences the progression of these diseases. So our sources of inspiration for the type of diseases that we look at are, we're interested in aortic valve disease, as well as several types of cancer, and dermal wound healing. I'm going to talk about the top two today, but first I'm going to focus on aortic valve disease. So in aortic valve disease, you start with this nice healthy trileaflet valve that's all nice and smooth, and it opens and closes with blood being pumped through it. But it becomes diseased, essentially gets fibrotic and calcified, and that makes it really hard to open, and also it makes it so that it doesn't close all the way. And so that creates problems with respect to blood regurgitating back into the heart, and it's not just an effect that then affects your valve only, it affects your whole system, because you can't pump blood as efficiently. And it's extremely prevalent. It occurs in about a quarter of individuals over, say, age 65. Not all of those people will need treatment for this valve disease, but it does occur to some extent in about a quarter of those people. And even if it's not severe, it is associated with an increase in mortality. So both from cardiovascular events and from any event. So again, sort of driving home this message of valve disease, even if it doesn't get really bad, affects your whole body. Risk factors are advanced age, being male, high cholesterol, and smoking. And the treatment for it is nothing other than total valve replacement. And that's where we see sort of our calling in this disease, is that you can see that a valve is starting to get diseased clinically, but then you can't do anything to stop it. And so aside from replacing the valve in the end. And so that's where we want to try to model the sequence of events in disease to figure out if there is a point in the disease progression that we could target to stop the disease from progressing. So If we can stop the progression, perhaps we can avoid the need for a valve replacement. The problem is we don't understand the progression. So there's a lot of work to be done before we can meet that goal. And one of the things that has hindered progress in this area is that most of the characterization of the disease features in valves has come in the end stage of disease valves. So you can get valves that are being removed from people during valve replacement surgery, and you can see a lot of things that are going on in these valves. You can see there's cholesterol deposition, there's inflammation in these valves, the extracellular matrix does not look right, you have blood vessels there when blood vessels normally aren't in a valve, the cells are proliferating, the cells are dying, you have calcification. But the problem is that getting that snapshot of late valve disease doesn't help us understand which thing comes first and which thing precedes a sequence that can be targeted to stop it from continuing. So, we have worked to sort of narrow down what is happening in early valve disease, because we think that there's a lot of thinking that once it gets really late in disease, it's probably past a point of no return. And so we want to see sort of at this stage where you can detect that valve disease is happening in a human, but hasn't gotten too advanced. We want to see if we can understand what happens in that time frame, so we can see if there's a target. And one of the beautiful serendipitous things that happened to me here in Wisconsin is that I was looking for something else on campus and happened to be directed to a person who has these pigs who develop really bad atherosclerosis, so vessel narrowing. And talking to this person, they're like, we've never looked at their valves. They might have diseased valves. And so I thought, this is fantastic. And sure enough, it was really cool. We got this opportunity to study these pigs that have this mutation that gives them familial hypercholesterolemia, which is a condition that occurs a genetic condition that occurs in humans. And these humans tend to get aortic valve disease at a really young age. And so it made it nice to be able to look at these pigs that were sort of age matched to a person that would be getting the same disease at the same age. And so from looking at these pigs, from that big long list of features that I listed on the previous slide, we were able to see that these three things stood out as happening the earliest in valve disease. So deposition of lipids, an increase in specifically one type of matrix component, and inflammation. But still, because it's only a snapshot, we weren't able to understand the sequence or how these related to each other. So again, the animal provided us with this snapshot, and now what we wanted to do is try to understand the sequence. And so you're probably thinking, well, you have these animals, couldn't you just, get them at different time points at different ages and get the sequence from there? So here's the problem. So there are other small animal models of valve disease too. The problem with those is that the valves don't really match what a human valve looks like, and they don't really get the human type of valve disease, and their valve extracellular matrix doesn't match what the human valve has. But we have these pigs that look pretty good, right? Okay, so here's the problem with them, is that they had to be between 1 and 3 years old to show disease, and at that point, the cost of keeping the pig is up to about $10,000 to $20,000 per pig. So clearly, this is not a sustainable source of disease tissues for us to look at and examine for causative relationships. So the conclusion that we kind of make from this is, okay, if we want to study disease tissues, we're going to have to make our own. And to give you an idea of what this looks like as a valve becomes diseased, so this is sort of a macroscopic picture where you see sort of nice, smooth valve. It starts to get a little bit sort of chunky here when it gets sclerotic, and you see it's thickening, and it thickens particularly in this one extracellular matrix component called glycosaminoglycans, or GAGs. And then at the end stages of disease, you see sort of these bony nodules forming on it, and you continue to have a different type of extracellular matrix disarray. And so we want to study this middle stage. We want to know, how did it get like this, and how can we stop it? And so today I'm going to talk about 2 different avenues that we've taken to make our own diseased valves. One is a top-down, so more traditional, or bottom-up, so more traditional tissue engineering approach, where we combine cells with a scaffold to make a diseased-looking tissue. And the other one is a top-down, where we actually start out with native tissue and try to make it diseased. in vitro. So I'll talk first about the bottom up. So our starting hypothesis was that this big change in the extracellular matrix that we see, we think that's going to be sufficient to initiate subsequent pathological events. Because I'm all about the extracellular matrix, and I think it does everything. So first we need to look at what extracellular matrix is in a valve. We have an assortment of ECM components, but we decided to narrow it down to the ones that seem to be the most prevalent in the valve and undergo the biggest change with the disease. So collagen 1 is sort of like the backbone of the valve. It has collagen 1 throughout. And then you have these glycosaminoglycans, or GAGs, and specifically hyaluronic acid and chondroitin sulfate are the GAGs in the valve. And these are also a very big component of the valve, both in the healthy times, but particularly in disease. These are what become enriched by about fourfold in this early disease. And then finally, our valves are primarily populated with valvular interstitial cells. These are a fibroblastic cell type that can differentiate into other diseased type phenotypes. There is also one other cell type in a valve, valvular endothelial cells, that coats the valve. And for the purposes of this investigation, we sort of ignored those for now, but they are very important. So what we did is, I didn't get into the nitty-gritty of how we made these scaffolds, but I can answer questions about that if you're interested later. It's also here in our paper. And so we made scaffolds that represented a healthy valve environment and a diseased valve environment. And the way that these differed is in the amount of GAGs, so HA, hyaluronic acid, or chondroitin sulfate that they had. And again, the hypothesis that we're testing is that GAG enrichment, so more GAG, we think is going to promote more disease. In particular, we thought it would promote inflammation. And so this is an inflammatory marker. And you'll see, as we had more GAG, we actually had less inflammation, or less production of this inflammatory marker. So we thought, well, we'll look at some more. We looked at other inflammatory markers. So this is another inflammatory marker. And again, especially with the chondroitin sulfate, we had less of this inflammatory marker produced. And we went on, we did a couple more inflammatory markers, and they all look like this. So if you look at our hypothesis and you look at this graph, what you realize is that our hypothesis was wrong. Increased GAGs are actually associated with lower inflammatory activity. And that's okay. And in fact, actually the story that ended up happening was way more interesting. So hypothesis being wrong is not always a bad thing. But it comes back to this issue of these valves had this huge increase in GAGs. And the body doesn't do that for just no reason. There has to be a reason why we have this fourfold increase in GAGs in these disease valves. So we want to know, okay, what exactly is that overabundance of GAGs doing? So we went back to our little triad here of our different events happening in early valve disease. So that one, those two don't connect directly to each other now. So we want to look at those other arrows and see if those are connecting. So what we first looked at are, okay, do the increased GAG connect somehow to lipids and the presence of lipids there? And this is now where we started to see something, not with the HA, but with the chondroitin sulfate. The more chondroitin sulfate we had, the more deposition we had of overall lipids, as well as in particular oxidized lipids, which are particularly powerful when it comes to disease progression and inflammation. And then we looked at this other connection between lipids and inflammation and saw that, indeed, when these valve cells receive oxidized lipids, which we just saw were trapped more by our diseased valves, we're getting more production of inflammatory factors. This is another one that we measured, monocyte chemoattractant protein 1. Okay, so now both of these are a yes. These are connected, and these are connected. So then we sought to do this sort of multi-step investigation of how can we put this all together? Can we actually create a platform where we can mimic multiple steps of disease pathogenesis in vitro, so in our dish still? And so what we did is we made our diseased ECM scaffolds, we incubated them with lipids and oxidized lipids, and then we saw whether those lipids got trapped by those scaffolds, and then whether those trapped lipids were able then to stimulate inflammatory responses by the cells. And in fact, that's exactly what we found, but in particular for the case of chondroitin sulfate, we'll come back to HA later, where we found that when you had 4x, so this diseased amount of chondroitin sulfate, it trapped oxidized LDL, And that oxidized LDL that it trapped, and to some extent the LDL that it trapped, stimulated the valvular and interstitial cells to produce more inflammatory factors. And not only that, but we found that that trapped oxidized LDL had a positive feedback loop where it signals the valvular interstitial cells to produce even more chondroitin sulfate. So start out with this sort of pathological amount of chondroitin sulfate, it traps lipids, and then it makes the system make more chondroitin sulfate. And so, of course, you want to check, is this real? And one of the questions that came up is that these inflammatory factors, most of the time people had assumed that they came from inflammatory cells that have invaded the valve. And so the question that we were getting at here then is, oh, do these inflammatory factors actually precede the arrival of inflammatory cells? So are the valve cells really responsible for producing these in early stages? And in fact, that's what we did find. So looking at our disease pigs, we found that, there weren't, it's in this paper, but there weren't any inflammatory cells here, but we did have big increases in inflammatory factors. So sort of verifying that, yes, the valve cells are responsible for producing inflammatory factors that are probably contributing to the recruitment of these inflammatory cells. But one of the other things, we noticed that the HA didn't seem to be doing much when it comes to lipid story. And one of the cool things about using these engineered platforms is that it can allow us to sort of separately study all these different extracellular matrix components. And so we could separate out the fact that it's not just overall GAGs doing something, but that chondroitin sulfate as a GAG is doing one thing, and HA as a GAG is actually doing a different thing. And what we saw it doing is that it's upregulating a molecule here that promotes a different aspect of valve disease, namely the growth of new blood vessels. And I mentioned this here because I'm going to come back to it in our next story. So this approach of being able to tailor these scaffolds allowed us to sort of individually look at the contribution of these different elements. Okay, so we started out with this. Well, actually, we started with a whole big clump of things and narrowed it down to these three. And then, but we still had these three that we were trying to sort of piece out. How are they connected to each other? And by using a fairly simplistic model of valve disease, we're really just varying this GAG content. We're able to take that triad and turn it into something where we know a lot more about the sequence. We know more about, we don't know how the disease ECM happens in the first place. We'll get to that at some point. But that some elements of this lead to factors that are involved in angiogenesis, that these diseased ECMs can trap the bad cholesterol, so oxidized LDL, which can lead to inflammation. I didn't discuss it here, but cell growth and cell death, which makes, and it also can lead to making more disease matrix, which can then feed back and make the scaffold more disease. And that's, overall, this yielded mechanistic data that we were able to confirm, again, with those scarce in vivo specimens. So using those in vivo animals or in vivo models as a validation step. Okay, but says reviewer number 3, it doesn't exactly match native tissue. And no, it does not. As I mentioned, I know it's a simplistic model. We've totally omitted a lot of things. But, reviewer 3 has a point, and how do we know that it's good enough? And so for that, we started on this somewhat ambitious goal of saying, okay, can we take the whole native tissue and make it diseased? And not while it's in an animal. We have this abundance of healthy valves available to us because people eat a lot of pigs. And so we can get from our local butcher, and there are lots of those in Wisconsin, healthy heart valves quite readily. But how can we make them diseased? And so that's where we had this idea of, can we actually do what we're calling ECM editing in ex vivo organ cultures and be able to overexpress these elements that we see in abundance in valve disease? So in particular, can we enrich our matrix in, say, the HA or the chondroitin sulfate that we saw in our bottom-up model? Okay, so here's the problem, though. We want to manipulate these GAGs, and it'd be easy enough, and actually we've done several years ago, ways to decrease the GAGs in valves. You can throw some enzyme on there, but how do you increase your ECM there? And really the only way we saw for doing this was to do some sort of, genetic overexpression. But the problem with GAGs is that they're not proteins. They're not primary gene products. And so what you have to do is you have to overexpress the gene for the protein that then synthesizes the GAG. So for the case of hyaluronic acid, you have to overexpress hyaluronic acid synthase and depend upon that getting made, and then that in turn making the hyaluronic acid. And then this hyaluronic acid must not only be synthesized, but then deposited and retained in our Valve ECM. And all this has to happen in the time frame of 3D organ culture and not disrupt any other ECM. So it's kind of a tall order that seemed a little bit nutty for a while. But spoiler alert, it worked. So, so what we have here is, I'll take you through the different steps. Actually, there are a lot of different steps here that I'm skipping, but sort of the main ones are, can we actually deliver virus to promote overexpression of HA, and we delivered HA synthase overexpression gene, in order to get more HA synthase produced that will then produce more HA. And so we delivered those to intact leaflets. Okay, so this is sort of a tissue that we had dissected out and we're keeping in just regular cell culture plate. And so yes, we get overexpression of our HA synthase gene. In fact, quite a bit overexpression. It peaks around day 3. So far so good. So now let's see step 2. Okay, we got the HA synthase. Did it actually go on to make any HA? And did that HA actually stick around in the valve? And so this is staining for HA, and you know, normal healthy valves will always have some HA in it. They should. And these are treatment conditions. This is a high titer of our virus. This is a low titer of our virus. This is sort of a sham one where it doesn't contain our HA synthase gene. And we quantify these with an HA ELISA. We found that with our high virus titer, we were actually able to get a 4-fold increase in our amount of HA in our valve, which was just really awesome because 4-fold, if you remember, was the target disease amount that we want to see for GAG enrichment to make a disease-looking valve. Okay, success so far. So next, you know, is this HA actually doing anything that it should be doing in a normal valve? And so we looked at a couple things. One, is it affecting the mechanics of the valve? So one nice thing about the biomaterial environment that we created is that we can control the stiffness and keep it constant. In a natural tissue, we can't do that as easily, but that in this case, it's okay. You accept some variation in other variables for the sort of fact that when you have more HA, it actually should be affecting the modules. This is actually really nice confirmation that having the HA enrichment was biophysically meaningful. And again, coming back to some of the results that we found from our bottom-up environment, where we saw that more HA caused our cells producing more VEGF, which is this angiogenic factor. And we saw that same thing in our top-down tissues. When we enriched in HA for our organ cultures, those cells started producing more of VEGF, which again, reminding you, this is similar to what we saw in our bottom-up environment. So it's really nice that now we have these consistent models with each other. And we confirmed that the production of this VEGF was specific to the HA being present. So if we blocked the cell's ability to interact with that HA, they didn't produce more VEGF. But when we had sort of a sham blocker, they were able to still produce more of that VEGF. So what are the functional outcomes? So it's one thing to say, you know, I have more HA present, it's making the cells produce more VEGF, but does that increase in VEGF, is that actually physiologically meaningful or biologically meaningful? And so what we did is we took our ECM edited leaflet, so where we had this enrichment in HA, and we took the media from that, so our cells in this leaflet are producing more VEGF and it's sort of going off into the media. And then we took that media and applied it to sex match, so these were male porcine endothelial progenitor cells, because we wanted to see, okay, the amount of VEGF that's now being overproduced, does it actually have an effect on endothelial cells? And in fact, it did. It made the endothelial cells proliferate more, so divide more frequently. And this is an assay where you're looking at the ability of endothelial cells to form network-like structures that could be predictive of their ability to form vessels. So as I mentioned, the formation of vessels in a valve is a bad sign. It's pathological. And we saw here is that this conditioned media from our HA-enriched scaffolds was able to promote more of what's called tubulogenesis than our control leaflets. And we could see that in the outputs when we started to measure tubes, is that our leaflets that had the enrichment of HA were able to form more tubes, and actually that we think this was directly due to the VEGF and not any HA that's in the media. So checking for physiological relevance now. So angiogenesis is a known hallmark of aortic valve disease, but this is the first evidence connecting it specifically to valve HA enrichment. So if we stain for VEGF in our piggies again, and our 1-year-old, which isn't very diseased yet, and our 3-year-old, we see a pretty big increase by our 3-year-old, which also had enriched HA. And one of the things that was unique about this is that there's a lot of literature out there about how HA can directly stimulate endothelial cells to be angiogenic. But here what we're actually showing is a different mechanism where HA stimulates an intermediate cell type, so our fibroblastic cells, our VICs, to produce an angiogenic factor that in turn can make the endothelial cells be more angiogenic. So we're really excited about these organ cultures as tunable biomaterials, the fact that we can take an intact tissue and overexpress ECM-related genes to get this selective enrichment in a certain ECM protein, or in this case, polysaccharide, and that this is both biophysically and biochemically relevant and functional to the function of our tissues. And so we're interested in exploring this further. You might also be wondering why we did HA when chondroitin sulfate was sort of the main part of the first story, and that's because we started this before we knew that chondroitin sulfate was the one that did more with lipids. So just the progress of science. We'll get back to chondroitin sulfate as well. So next generation of these disease models, what can we do better? One of the things that we're really interested in is cell sex. So male versus cells derived from males versus females, they behave differently in valves. So when we take those cells out of those valves, and even if we leave them in and do some gene expression profiling, we find that the male cells in general seem to exhibit more pathological markers than the female cells. And so we sex separate all of our cell cultures. I know that a lot of people think about clearly looking at full organisms, male versus female animals, male versus female, female humans, but that on the cellular scale as well, it matters what the source of your cells were. And some interesting data from the field of heart valve disease too is that men and women may get different types of heart valve disease as well. So for the same severity of disease, it was found that men actually had more calcification and women had more fibrosis. So we're really interested in seeing if we can understand where the sexes diverge in their progression of valve by modeling across the whole valve disease continuum. So making not just our simplistic sort of healthy versus early disease models, but making healthy, early disease, late disease, male and female models to understand whether we can get at stage and sex, biological sex-specific targets. And some early evidence from that it does matter are that we may have made some of these healthy early and late models and cultured female versus male derived cells in their valve cells. And we looked at a marker of, this is a marker of our cells assuming sort of a pathological phenotype. And we found that our male response to pathological extracellular matrix is 2 to 3 times that of the female. So we are again seeing a differential sensitivity in the ability in our cells based upon the sex of the animal that they came from. Okay, so you might be thinking, that's cool, but I don't really care about valves. A lot of people don't think about heart valves in their everyday lives, and that's totally okay, although it is really important disease. But I'll spend these last few minutes just talking briefly about breast cancer, which is something that more people tend to be a little bit more familiar with. So breast cancer, sorry, my stats are US specific. It is the most common cancer in US women. There are about 42,000 deaths per year in the US due to breast cancer. When it progresses, what happens is that tumor cells invade tissue and then metastasize to other sites in the body, and that's where you sort of end up in trouble. And the problem is that eight out of 9 drugs identified in animal models sort of fail when it comes to being implemented in the clinic. So clearly there's a need here to develop better predictive ways of analyzing what drugs are going to be successful. And we think that using human tissues in the lab in these tissue engineered models can help us get to that place. One of the questions that we're interested in is the fact that stiffer tumors tend to be more invasive. So if you have a stiff tumor, the cells are more likely to metastasize elsewhere. And we want to understand what's responsible for that increased malignancy of those stiffer tumors. And so what we did is we made these biomaterials, and they're a little bit different from what I was using in the Valve materials, and you can see the details of the materials in this paper. But basically, it has a collagen-based backbone. And what we've done here, though, is we've used collagen in a couple of different forms. We've used collagen in a gelatin form, so that's denatured, so we can control the stiffness. And we've used collagen in its native form, so we can still have fibers. And we did that because fibers have been shown to be important for breast cancer or for the invasion of multiple types of cancer. So we wanted to make sure we had those fibers present. We didn't actually understand how important they would be until we started doing this study, though. We thought they just sort of helped to facilitate certain cellular events. We didn't realize that some cellular events wouldn't even occur without the fibers. But in fact, that's what happened. So here we have spheroids of breast cancer cells. That's what these MDA MB231s are. I know it's a long and funny name, but just think of these as breast cancer cells. And what we had here is different stiffnesses. So this would be soft, medium, and stiffer, and different amounts of fibrillar collagen. So they still have the same amount of denatured plus fibrillar collagen presented to them. So their protein presentation is constant. They should have a constant ability to interact with sort of like the adhesive and degradation sites of gelatin and collagen. But we changed essentially the amount of fibers available to them. And one thing that struck us immediately is that if you didn't present these cells with fibers, they would not migrate out of the spheroid. They could not invade their surrounding areas. And the other thing that was important here is that their invasion into their surrounding areas decreased with increasing stiffness, okay? And this didn't seem to be dependent upon fiber density. It was sort of a yes-no or off-on with the fibers. If the fibers were there, then they invaded. If they weren't there, they didn't. But once the fibers were there, it didn't affect extent of invasion. Okay, so here's where we get into our problem, though. What's consistent is that collagen fibers are important for both in vitro and in vivo. What's not consistent is that when we did our in vitro model, we got decreasing invasion with increasing stiffness. But in vivo, it's the opposite. When you have increased stiffness, you actually get increased invasion. So we thought, is this just a terrible model, or is there something else going on? And you sort of, cross your fingers for the second option there. And so we took a closer look, and we took a look more at the kinetics of how the cells looked as they were invading. And what struck us is the invasion kinetics. So this is our low stiffness across the top. This is our high stiffness. You can see low stiffness right off the bat. It is going out there, it's invading. High stiffness is not really doing much, and then bam, it really starts to ramp up. And so I thought, well, this is potentially really interesting. And also, just to verify, without any fibers, there's no invasion. So what's going on here with these invasion kinetics? Let's look more closely at these cells and see how they're differing. And so we started to zoom really closely in on these cells. We started to notice that, the cells that are invading, they also just look a little bit different in the low stiffness versus the high stiffness. We noticed a lot sort of more like jaggediness of our high stiffness, of the cells invading in the high stiffness condition. And so what we did is we treated these conditions with an MMP inhibitor. So that's matrix metalloproteinase inhibitor. And what this does, matrix metalloproteinases degrade collagen and lots of other things, but in this case, collagen. And so what we were doing here is we were stopping the ability of the cells to sort of, degrade the collagen that's in their path. And interestingly, what we found out is that in the case of low stiffness, the cells still invaded. But in the case of high stiffness, all invasion was quashed. And so now that starts to tell us a lot more about the types of invasion going on here. In our soft scaffold, it seems to be more passive. So cells seem to be just sort of, like groovy, meandering out of the spheroid. But the stiff scaffold, they have to work a lot harder to get out of there. They adopt this different morphology. They have the more invatopodia, and this is entirely dependent upon their ability to degrade the surrounding matrix. Coming back to the ECM, we found it's dependent upon another thing too. When we were looking at what's going on ECM-wise in these scaffolds, we identified fibronectin, which is another ECM component, as being super high in our high stiffness scaffolds. This is sort of a heat map of fibronectin around our spheroid here. And you can tell they're really intensely producing a lot of fibronectin only in the high stiffness scaffold. And in particular, it's a type of fibronectin known as EDA fibronectin. And it was only being produced in high stiffness. It wasn't being produced in low stiffness at all. And so we did, you can actually check out our paper that was just published earlier this year and read more about it, but we did multiple experiments to knock down this fibronectin production and found that in fact it is absolutely essential in the high stiffness environment that these cells lay down this fibronectin in order to invade. And so this was really cool because it was uncovering a mechanism of you're in this high stiffness environment, and essentially what's doing is it's promoting potentially more malignancy by having the cells be more aggressive in their invasion in the surrounding area. So the cells had to be super aggressive. They had to be very, focused on degrading their matrix out of there, laying down more matrix so they could invade out of there. So this now could help explain why stiffer tumors are more malignant, because those cells are actually adopting a different phenotype that makes them a little bit more fierce. But also, why does this matter? We now again went back to our validation step and looked at human tumors. We looked at this tumor microarray, so from 200 patients or so, it's little sections of their tissue, and stained for this EDA fibronectin, so pictured here in green, in benign versus triple negative breast cancers. And indeed, we found out that this marker that we saw was necessary for invasion of these stiff scaffolds was also elevated in these highly invasive and malignant tumors. So resolving our initial contradiction, stiffness initially attenuates invasion, but then activates pathways to make the environment more permissive to invasion. And that this particular ECM component, EDA fibronectin, is essential for that invasion to occur. But sort of most of all, none of this would have been possible for us to understand without fibrous and mechanically tunable scaffolds. So closing up my talk here, so the extracellular matrix is a great thing. It's really it's a great thing, but also could be a bad thing. It's a great thing to study because it's just really fascinating and plays such diverse roles in cell behavior. And it's really influential in driving disease. We've found ways to introduce it into intact native tissues, and that these disease mimicking models can offer us insight on how diseases are happening. And that it's not just extracellular matrix identity that matters, but it's also its topography and its biophysical aspects that really matter too. So our vision for our lab is that we want to use these models to identify new treatment targets. We want to reduce the number of research animals used, and we want to improve the drug hit rate so that, with our hypothesis being that human tissues can be a better predictor of human drug response.
All right. Well, I think you're muted, Kristyn.
Am I back now?
Yep.
That was weird. I did not hit anything. Okay. Can you see my presentation? Okay. So thanks to this really awesome team of people who did all of the great work. So that is the end of my talk. I am happy to take questions.